Hello, hello, hello. How are you, everybody? Here I am, Sharmalak Wester, biologist here at SUS Herman Minor School, Hawassa. And as usual, we shall proceed our lecture. And today I'm coming with another new topic from Unit 4 Biology that talks about evolution, of course. Particularly, we have been talking about one aspect of this, one subtopic of this, one cause of evolution, that is mutation. We have been talking about mutation, specifically the first type, that is the point mutation, which involves the change in one nucleotide, which usually happens during DNA replication, of course. Today, we shall see the growth change. The growth change, that means the chromosomal mutation. When the whole chromosome or the part of chromosome is missing, or part of chromosome is getting change or alteration or change in the chromosome, which is of course much more huge change as compared to the point mutation, and the consequence is also that much more serious. Therefore, we shall talk about chromosomal mutation today. Please don't forget subscribing and liking, sharing my videos, so that you will get my newly made videos. And please give your comments also, so that I can improve my lessons or lecture. Let's go now. A chromosomal mutation is a mutation involving growth changes in the chromosomes. You see, we are talking about change in the chromosomes. What's chromosome? Chromosome principally is composed of nucleic acid DNA and histone proteins, special proteins which used to hold the DNA. Histone proteins are proteins, special types of globular proteins on which DNA would surround, and so that it is kept organized, especially during cell division. Therefore, the whole change, the DNA plus the whole growth change, the chromosomes, the DNA plus histone proteins as a whole, is what we call chromosomal mutation. That is, it involves a long segment of DNA. Yes, long segment of DNA. Chromosomal mutation occurs most often during meiosis. If it is error in it is error in meiosis. If there is error in meiosis, which means formation of reproductive cells in sexually reproducing organisms, and this change includes one, during crossing over. This is crossing over is a natural event where during meiosis one and prophase one of meiosis one, homologous pairs of chromosomes come near to each other in a process known as synapses, and they exchange part of their lower arms, so that the increase crossing over event increases genetic diversity in the population. The second process that causes chromosomal mutation is, of course, crossing over causes structural chromosomal mutation. The second process that causes chromosomal mutation is non-disjunction, that is failure to separate. Either homologous pairs fail to separate, or sister chromatids fail to separate during anaphase stage of meiosis. Be careful, during anaphase 1, homologous pairs of chromosomes fail to separate, whereas during anaphase 2 of meiosis 2, sister chromatids fail to separate, so that there is chromosome number difference, either difference in 1 or plus 1, plus 2, plus minus 1, minus 2, plus these changes. And also, of course, there is another type of chromosomal numerical mutation where the number of chromosomes change it in such a way that the chromosomes increase by the number n. The number n. This is naturally done, especially in plants. Naturally done in plants. Plants increase their chromosome number by ploidy. Ploidy means n, so that from 2n they go to 3n, then 4n, then 5n. This is polyploid, of course. It's not connected with non-disjunction or crossing over event here. Crossing over event, principally crossing over event, causes one type of chromosomal mutation, which is known as structural chromosomal mutation, whereas non-disjunction causes one type of chromosomal mutation, which is numerical chromosomal mutation. From the numerical chromosomal mutation, specifically, we come to that point, specifically it causes aneuploid, where a cell is with one extra or two extra chromosomes, or deficient in one extra chromosome, or deficient in two chromosomes, not more than that, of course. Let's go. Types of chromosomal mutation. I have already told you. Chromosomal mutations are studied under two broad categories, of course. One, chromosomal mutation type 1, which is also, as I've told you, structural change, structural chromosomal mutations, where a chromosome undergoes structural change. The second is chromosomal mutation type 2, which is numerical chromosomal mutation, that is the number, deficiency in number or surplus in number. Let's discuss the first one, chromosomal mutation, which type 1, which is known as structural chromosomal mutations, is also known as structural chromosomal aberrations. Change in chromosomal morphology or structure, also known as chromosomal aberrations. These are much bigger events than point mutation and usually cause death of the fetus or the embryo is aborted. So, such mutations commonly are referred to as lethal mutations which can kill or which do not allow the embryo to complete its embryonic development and be born safe. You see? In a chromosomal aberration or structural change, there is an alteration in the structural organization of the chromosomes. The structural changes could be one, deletion, when part of chromosome breaks and is lost. Two, inversion, when part of chromosome is broken and rotates 180 degrees and reattaches that particular site. C, translocation, very important, translocation to bring about evolution, very important this one. Translocation is when chromosomes, non-homologous chromosomes, cut and exchange their parts. Deduplication, when part of a chromosome is found twice or more. Duplication, redundancy of the chromosome. Let us see each one by one. Deletion, part of a chromosome may break and get lost. You see, this is serious, depending on the size of the broken piece of the, as the size increases, the damage, the loss is huge, so that this is serious as far as mutation is concerned. Part of a chromosome may break and get lost. And the deletion could be, terminal deletion. One piece breaks off from one end and is lost. Terminal. You can see this part is losing, the whole this part is losing. This part breaks here, and therefore terminal, from one end. A chromosome piece is broken from one end, is lost. The other is interstitial deletion. This occurs when a chromosome breaks at 2 points with its arm, 2 points within its arm, and the segment between the breaks is lost, while the remaining ends of the chromosome fuse together. You can see here, this part is lost. You can see here, this part is lost, and this is known as interstitial deletion, of course. Again, interstitial deletions can vary in size from small micro deletions, very small size very important, from small micro deletions to larger segments, and the effects depend on which genes are lost by the deletion, of course. The larger the size lost, the larger the number of genes lost. That means the larger its effect is. When we say microdeletions, very, very tiny part is being lost. In microdeletion, of course, still microdeletions occur when the deletions in the chromosomes are too small to be detected, but it can result in a loss of several genes and cause a range of genetic symptoms. Still, even microdeletions, depending on the gene, the importance of the gene that has been removed by that piece. Importance. It depends the effect of microdeletion. However, it is very small part of the chromosome is being lost. The type of the gene lost, the types of gene lost matters. I can give you a particular example of this microdeletion, which is relatively common. Microdeletion, the YCM microdeletion, they call it YCM, which means Y chromosome microdeletion. Y chromosome, you know, the sex chromosome, one of the sex chromosome. It's very small, still in that small part of the Y chromosome, which determines principally maleness. If microdeletion occurs in specific points, Y chromosome and microdeletion refers to the loss of small segments of DNA on the Y chromosome, which can result in, of course, this causes azoospermia and oligospermia. Azoospermia refers to absence of sperm. You can't produce. If you have Y chromosome microdeletion, you will suffer from azoospermia or absence of sperm or an inability to produce sperm cells, or oligospermia. You may produce a very small amount of or number of sperm cells. Therefore, your fertility reduces. In azoospermia, you lose your fertility totally. In oligospermia, your ability to inseminate, ability to fertilize the egg of the female, your fertility is partially lost. Again, the second type. Now we have seen the first relation. The second, inversion. An inversion part or piece of chromosome breaks off, then turns 180 degrees and reattach in that site. When this doesn't include the centromer, you see, inversion can be divided into two. When the inversion part of the chromosome that is broken and rotates 180 degrees and reattaches, when this doesn't include the centromer, it is called the paracentric inversion. You can see from this diagram, paracentric, you see, the centromer is not involved in the broken piece. Only the upper arm, you can see in this case. It is broken there, rotates 180 degrees and reattached in that same place. This is paracentric. The other is when the inversion does include centromer, it is pericentric. You can see this diagram. You see, very big portion is broken, rotated, and which includes the centromer at the middle position here. Pericentric inversion. Pericentric inversion is much more serious than the paracentric inversion. Now, the third is translocation. Very important. This one is very important as far as evolution is concerned. Of course, translocation happens when a segment of a chromosome breaks off and then relocates to a completely different chromosome. Completely different chromosome. And this creates fusion chromosomes, fused chromosomes. You can see, for example, in humans, chromosome number 1 and chromosome number 2, which are totally non-homologous chromosomes, different chromosomes, may cut their parts and exchange. Therefore, this creates fusion chromosomes as one type of chromosome fuses with another and may lead to various syndromes. Translocations can be classified into two main types. You can see here. One, reciprocal translocation. One way to classify translocations is into reciprocal translocation, and we also have a special type of translocation known as Robertsonian translocation. When I come by the next lesson, I will start from this portion and proceed. We will continue with the next portion. We shall discuss one by one reciprocal translocation and Robertsonian translocation, plus other aspects of chromosomal the structural chromosomal mutations, including duplication, the last one. Thank you very much. I will come by the next portion of this unit, this specific part of the unit. Thank you very much.